Since 1665, we have understood that organic systems, like us humans, are made up of microscopic cells, thanks to Robert Hooke. Normal cells in our body serve a particular purpose. For example, a heart muscle cell's function is to contract and expand, allowing our heart to pump blood. It has a specific purpose. But it's only been in the last 30 years that scientists have developed an understanding about a cell that doesn't have a specified purpose, as it can become any cell type. This universal hero of the human body, founded in 1981 by Dr. James Till and Ernest McCulloch, is called the stem cell. The stem cell was founded through embryonic mouse tissue. In today's research, stem cells are still obtained from human and animal embryonic tissues, or fetuses. Stem cells can be sourced by other means, such as our fat cells. Recently, we even found stem cells inside our teeth. Stem cell collection from fetuses has continued to gain controversy, as it requires embryos from women that have undergone abortions. In essence, the research benefits from the destruction of these embryos. Due to the continual ethical discussion over the sanctity of life, this type of stem cell research, although practiced, will continue to face scrutiny. Do you think embryonic stem cell research is ethically wrong? There are embryonic stem cells and there are also adult stem cells. Scientists can also identify stem cells based on their potency. Totipotent is the most potent, which means it can form a fully living person. The best example is the fertilized female egg. Pluripotent can develop into any human tissue or organ. Multipotent, or the adult stem cell, can alter into a specific range. And unipotent can only become one cell type. Muscle stem cell equals a muscle cell. In recent scientific research, stem cell therapy has become a strong focus in neuroscience, especially relating to Parkinson's disease, or PD. According to the Parkinson's organization, sadly 10 million people suffer from PD worldwide, and there is still no treatment to stop the progression, although there are treatments to relieve symptoms. So, what is Parkinson's? Parkinson's is a neurodegenerative disease, which is where neural cells in the nervous system degenerate and die. Unfortunately, the cells in the nervous system find it difficult to regenerate after their death, leading to permanent damage to motor functions. What's also sad is that Parkinson's gets worse with age. Stem cell therapy's role in Parkinson's treatment is to slow down this degeneration and replace the cells, allowing the repair of the nervous system. At a closer glance, Parkinson's is caused by the decline in dopamine in the midbrain, due to the loss of those neural cells I just mentioned. You may have heard of dopamine. It's a molecule that makes us feel good, but it also sends messages through the nervous system. Basically, it lets our cells communicate. A disturbance in nerve communication due to the lack of dopamine causes trembling in one's arms and head, impaired balance, stiffness in the muscles, and loss of motor function. The loss of dopamine in the early stages of Parkinson's can also lead to depression. Although there is still no reliable stem cell treatment for Parkinson's, many scientific studies on animals and some human trials have shown success. These studies focus on regenerating those dopamine molecules in the brain. Research conducted hasn't focused on just one type of stem cell either. For example, one type that has been researched is the fetal neural stem cell, or FNSC, which is transplanted into the patient from fetal brain tissue. In the last decade, scientists have genetically developed and researched another new stem cell called an induced pluripotent stem cell. In essence, these IPSC cells are adult stem cells taken from a patient's blood and are genetically reprogrammed to act like an embryonic stem cell, meaning it's of the highest potency. These IPSCs are perfect for Parkinson's research, as they bypass any possible rejection of transplant material, since the IPSCs can be made from the individual's blood. iPSC cells have shown massive success in animal trials, where tremors and motor function have improved. If these iPSCs continue to prove reliable and therapeutic effect for future patients, embryonic stem cell collection may become unnecessary. However, for now, embryonic stem cell research is continued, as there is still uncertainty concerning iPSCs. There are significant implications with all types of stem cell research, including the use of iPSCs. The risk of tumor formation from stem cell grafts resulting in replication of cancerous cells poses the most considerable risk and hurdle in the hopes of a reliable procedure. Although in many animal trials there's attempts to scan for tumor formations and prevent the risk, there is a need to test larger animals that have longer lifespans with iPSC cells to ensure no long-term cancer risk for humans. Other obstacles that prevent stem cell treatments from being available consist of understanding the suitable dosage of stem cells for patients, learning the most efficient way of dopamine release, and understanding the correct injection spot for patients. It is only through further research and more clinical trials that we will live in a world with reliable and successful stem cell therapy for Parkinson's. And that's all us viewers can try and do, is hope that the smartest and brightest in the neuroscience field, better yet, all fields of life, strive to do their best to improve on what we have now. If you feel inclined, use these references to do some further reading, as well as these 3 websites. These 3 websites also give you the opportunity to donate /find out more. Thank you for watching.